All right, last but most certainly not least, we have Nicole Anderson and McKenzie Scanlon from mathematics, and they will be presenting on predicting RNA structure using mathematical models.
Hi, I'm Nicole Anderson.
And I'm McKenzie Scanlon.
And this semester, we're doing research with Dr. Riehl in predicting RNA structure using mathematical models.
So many disorders and health problems such as autism and anaphylactic food allergies and some kinds of heart disease have genetic and epigenetic causes. And even considering just these 3 disorders alone, they end up costing the U.S. health system over $250 billion per year. In order to better understand the chemical processes behind these disorders, it's important for us to understand the function of certain stretches of RNA. So RNA, like DNA, is a molecule made up of nucleotides. You can just think of the nucleotides as the building blocks of the RNA in this case. So DNA is a double-stranded molecule, while RNA is a single-stranded molecule. So in RNA, the nucleotides bond together to kind of form this helix shape, and they make up what's called the backbone of the RNA. And the backbone of RNA is rather flexible, so it's able to kind of fold in on itself and form new hydrogen bonds between the nucleotides in the RNA. And understanding RNA folding is important because how an RNA strand folds significantly impacts the function of that RNA strand.
So you may have heard that protein folding is a very difficult problem. RNA folding prediction is actually much harder because RNA's backbone is more flexible than a protein's. Even on a segment of 100 nucleotides, there's the potential for more than 2 times 10 to the 78 different bond attractions. And to put a number this big into perspective, that's 2 times as many atoms as there are in the universe. So imagine you were holding one end of a string and you were to drop that string on the ground. We expect that string to land in kind of a heap, maybe touching itself in some spots, but we don't expect it to land in a perfectly horizontal line or that it's going to knot a bunch of times while it falls. So what we do is we eliminate these unlikely foldings from our model.
So what we did is look at theoretical graphical models of RNA strands, the ones that significantly restrict the unlikely structures from appearing. So we first looked at smaller strands of RNA and finding a way to classify the ones that were most likely to occur in nature. And once we were able to classify those, we were able to build larger RNA strands from the smaller RNA strands. And what we ended up doing was finding all RNA secondary structures that 5 different models have in common. And we were able to look at the growth rate of this common class and then also predict the size of this class as well for any size of or any length of RNA. And understanding the size of this class is important because it helps us to better predict how RNA will function.
Global human health problems are very complicated, and using mathematical models help us bring them down to a scale that can be analyzed. Solving the mysteries of genetics and epigenetics requires computer scientists, biologists, and mathematicians to all contribute. Chancellor Jim asked, what is our next step in our research, or what's the next stage now that we've done our first part?
So we looked at the intersection of a certain family of RNA, and so the intersection of all those different families of them, but then there's different families within those, so finding a way to classify those different families is something that we might be interested in looking at in the future.
Second place, Science and Technology, is awarded to Nicole Anderson and McKenzie Scanlon.